So once again, hello everyone, and a very warm welcome to all of you for joining us today. My name is Anuja Bhardwaj, and I will be your moderator and host for today's session. Before we begin, I would like to share a few quick housekeeping notes. This webinar will run for approximately 50 minutes, followed by an interactive Q&A session at the end. We encourage you to make this session engaging by submitting your questions or comments at any time using the chat box located on the right-hand side of your screen. We will address as many questions as possible during the discussion segment. Please note that all participants are currently in listen-only mode, and today's webinar is being recorded. The recording will be shared with all the registered participants after the session. To get us started, our discussion today focuses on accelerating FDA Fast-Track Monoclonal Antibodies through integrated strategy. As the biologics landscape continues to evolve, development teams are increasingly challenged to accelerate timelines while maintaining quality, regulatory compliances, and manufacturing readiness. In this session, our speakers will share the perspectives on how an integrated approach across process development, technology transfer, and manufacturing, quality, regulatory functions can help streamline the journey from development to cyclic GMP production. We are privileged to have with us a distinguished panel of experts from Aragen's biologics team, bringing extensive experience across biologics development, manufacturing, process scale-up, technology transfer, and quality compliance. Let me introduce our speakers of the day. Our first speaker is Indranil Sanyal. Indranil is Associate Vice President and Head of the Development Group at Aragen Biologics, Bangalore, with over 30 years of experience in biologics process development across both biopharma companies and CDMOs. He brings deep expertise in end-to-end biologics development from gene to clinic, including process development, scale-up, technology transfer, process characterization, and commercial manufacturing support. Indranil has a strong track record in accelerating development timelines through phase-appropriate strategies, risk-based approaches, and innovative process solutions, helping advance biologics programs efficiently from early development to GMP manufacturing. Our next speaker is Prithvish Dey, who is Journal Manager, MSAT, and Process Development at Aragen Biologics, with over 19 years of experience in biologics process development, MSAT, and cyclic GMP manufacturing across product companies and CDMOs. He has extensive expertise in process development, large-scale technology transfer, commercialization, and life cycle management with a proven track record advancing 15 plus biosimilars and novel biologics and executing 20 plus technology transfers up to 20 KL. Prithvish also brings strong regulatory experience supporting global submissions and inspections. Our next speaker is Abhishek Bhatt. Abhishek is Deputy Manager MSAT at Aragen Biologics with over 10 years of experience in biologics purification and process development. He has extensive expertise in the early and late stage development of therapeutic proteins and monoclonal antibodies with a strong focus on purification, process design, technology transfer, and scale up. Throughout his career, Abhishek has successfully led multiple tech transfer and scale-up programs, supporting the efficient advancement of biologics from development to manufacturing. We also have with us our next speaker, who is Gokul Ram. He is Associate Director and Head of Quality Control and is also a certified ISO 9001-2015 lead auditor. With over 22 years of experience in biotechnology, quality, and compliance, his expertise spans analytical method development, technology transfer, method validation, bioassays, microbiology, and facility qualification. Throughout his career, he has supported quality and compliance initiatives across biologics development and manufacturing and has handled audits by major global regulatory agencies, including US FDA, EMA, MHRA, and VISA, WHO, COPFIRIS, and Armenia regulatory authorities. With that, I would like to invite our first speaker of the day, Indranil Sanyal, to take the session forward. Over to you, Indranil.
Thank you, Anuja, for the introduction of the speakers. Really feel privileged to be actually interacting with the August audience. And today's topic, of course, is our own experience on accelerating a fast-track designated therapy of a monoclonal antibody. Of course, the examples and the strategies are not only applicable for the FDA fast-track molecule, but could be also implemented onto any other type of programs as found deemed suitable. With that, I actually just begin my slide. I go to the next slide, which basically talks about our motto. We are a CDMO service provider, and basically it's always the client comes first for us, and it's your science, and we have the solutions for that. And we together integrate both the science and the solutions to bring in better lives to the unmet medical needs. So I would give an overview about the fast track designated therapy. And as I said, it is not necessarily on the fast track therapy, but it can basically go to other antibody development or any other biologics. So that has to be taken, and I have mixed the strategy. What are the strategy in Aragen we take to actually do this fast track development for a fast track biologics or a normal biologics. This is briefly about our infrastructure. On the left-hand side is our laboratory located at the Morgan Hill site. They are expertise on cell line development and up to the preclinical development. That's where it is located at the California site. And on the right-hand side is the facility from where I belong, actually. And this site is actually having expertise on clinical development up to the GMP manufacturing. A quick round of services that we offer from our biologics facility. It starts from the cell line development and goes up to the GMP manufacturing. All interpressed modules can be done as an a la carte basis or an end-to-end program. So the right-hand side one shows about our capability as an integrated service provider. As I said, the cell line development and any other related development in the early phase of the work happens at the Morgan Hill facility. And subsequently, there are overlapping works that happens in the Bangalore facility, which leads to clinical manufacturing. All our manufacturing setup is a single-use system. Right now, we have capability to go from all the way from 50 liter to 2,000 liter GMP manufacturing. We are increasing our capacities over a period of time. We are adding up multiple number of 2KL bioreactors very shortly, and we are augmenting our drug substance capability with the drug product facility, which is also coming up very shortly. Nevertheless, as a service provider, we always keep some capacities open for an expansion, and we have a capacity of roughly around 15,000 liters at the rate of 3 bioreactors at the 5 scale. That's in the plan. The fallow space is available, and that would basically take a very short turnaround time to actually put those bioreactors and the facility in place. Basically touching on in our presentation today from some of our speakers out here. One of the key services related to the process intensification on the upstream side, morely targeted towards the improving the productivity and in a process improving on the cost of goods. On the right-hand side panel, towards the bottom side, we also work on optimizing the cost of goods. We have a very well laid down cost of goods model that is available with us in terms of optimizing the cost going towards the materials. So this would be also a part of a discussion. One of the speakers is going to talk about it, and both intensification and improving on the cost of goods would actually together help to bring down the overall cost of the production. With that, I move on to the next slide. Okay, this is a little busy slide, but I would like to spend most of my time in my talk on this particular slide. It basically shows up the strategy that we take around. Now, let's be clear on one part of it is that the misconception of FDA Fast Track necessarily does not mean that it allows to actually jump on processes and basically get approval. FDA Fast Track means it creates an opportunity for a significant, frequent interactions with the FDA through communications, through a periodic review, and on a rolling basis. And typically, these fast track therapies are given to medical needs which are unmet, or therapies that are already available into the market but have been found to have a lot of safety or toxicity issues, and there is a therapy you are developing which can alleviate all these things. So on those cases, there are potential chances of getting the FDA Fast Track designation. Now, what it means is that when it creates an opportunity, how we grab that opportunity. Now, the opportunity could be used not only for the FDA Fast Track therapy, it can be used across any type of a biologics molecule that we do. Here is an example of what we have done and what we do and how our strategy is. So coming to this particular Gantt chart, for anything that we start with, of course, the cell line development is the first activity that it happens. So from serial number, row number 1 to 3 are all cell line related development. The point here to note here is that depending upon the cell line development, what we do here is that we don't wait for the complete cell line development to be over or the availability of the research cell bank. We have cell lines and we have our platform cell lines, which basically helps in getting quality and actually behaves the same way as we would expect and once the RCB is produced. In other words, the quality produced by the stable pool, which we generate during the cell line development as the first milestone, we use that stable pool to start the process development work. So the row number six, which is basically the starting of the development work, which is an upstream development, which starts just after availability of the stable pool. Now, using the stable pool, we start the upstream work, and subsequently, on the way, we produce some quality materials which are fit enough to start the analytical development work. So in a way, we are not losing on the time. We are trying to use all the opportunities we have. Of course, nevertheless, it is important to note over here, when we say the development work, we have platforms for upstream, downstream, analytical, and even the workflow in our cell line development is a very well-established workflow. As you can see in row number 8, which basically starts soon after the analytical development, a portion of the analytical development have already started, and the downstream starts using the material of the upstream well ahead of time rather than waiting for an RCB. If you see that row number 8 also extends beyond the stable pool all the way into that. If you compare with row number 2, which is basically ending up in 16 weeks time, you would see that the downstream continues to happen. That means it gets to see RCBs also while the development are happening. Now, the reason behind actually starting all this together is, of course, to encash on the opportunities, but it allows us, if there are any surprises associated with a specific molecule, that can be tackled ahead of a time in terms of the timeline point of view and in terms of any kind of a tweaking that would require during the course of a development. Now we go to the row number 10, which is very important. In most of the cases, we wait for materials to start the formulation development work. But usually what happens when we do that, we know that formulation development is related to inception into stability. It takes certain period of time, and then basically, you get the results and then augment it with the downstream, probably the last unit operations of the downstream. Typically, in the biologics field, most of the time it would be a formulated drug substance that actually goes as a drug substance storage. So what we do here is that we use the formulation platform that we have. We have set of buffer mix, set of excipient mix that we actually screen, and in most cases they work very well, and that we augment it early as soon as the material from the downstream is available. Now what it translates into is that now we will compare between row number 9 and row number 11. What happens in that is that when we have done the development, when the development is going on, at some point of time, we have to integrate to see the process performance between upstream and downstream. What is the type of the recovery? What is the quality we are getting? And also that's the time we also generate material for the tox trial. Now, what it helps us is that once we have those materials generated for the tox supply, I would go to row number 11, which is very, very important. We can set up a non-GMP drug substance stability, which is basically a formulated drug substance in a representative vials. What it allows us is that by the time we will have the GMP material ready, we would have data of approximately around 2 months period of time, because it's very important that the clinical dosing is supported by a certain number of months, typically between 1 to 3 months is what is an ask. Around the same time, what we do here is that with row number 9, when we are generating material for tox, we also use this material for developing the scale down model for the virus clearance study, which is row number 13. And also it helps in basically doing a scale down model study, establishing the scale down model, because that has to be done earlier on, because for us, the virus clearance is an outsourced service. We have to establish the baseline runs. Now we have platform virus clearance scale down models that we typically use, and on a case-to-case basis, we have to tweak our process. Now let's move on to, well, all these things are happening. What is happening on row number 4 and 5? That's where the material for the master cell bank would come from. Row number 3 has finished with getting the lead RCB clone. Take that RCB clone, we start preparing the master cell bank, which is row number 4. And we have in-house testing. Of course, the characterization or the regulatory testings goes to an outsourced party, which is used for filing, and the in-house test is not used for filing. The in-house test is mostly directed towards de-risking the GMP facility before we expand a non-characterized to a GMP facility. We test for adventitious virus, we test for mycoplasma, we test for certain other attributes in order to de-risk the facility. Now that we have an MCB ready with us, we have rest of the things follows up, which is row number 13, 14, 15, and 16. Sorry, row number 14, 15, and 16. The drug substance manufacturing and the drug substance, clinical drug substance, which is being manufactured, is basically used for establishing the last part of the scale down model, and the material from this is sent for the virus clearance study. The drug product manufacturing also actually happens. We currently outsource this. The drug product manufacturing happens, which is in row number 15, and subsequently we release it at our end. So row number 15 constitutes both manufacturing and the release, and they get inserted for the stability. So now, if we compare between row number 11 and row number 16, we already have the 2 months stability data. We potentially would be actually having initial outcome of the virus clearance study based upon our repository of the data that we have. Now, this is an overall working in together, multiple modules getting activated, and as a result of we achieve the speed, we deliver it on time, which is typically in the range of 8 to 9 months of time. So the nutshell, the message is, as it reads here, fast track designation would create an opportunity. How we use that opportunity is very important, and that can be used by using the integrated execution, starting with the stable pool, platform process, early analytics, formulations, and removing the bottlenecks like some of these outsourcing services that are there. With that, I move on to the next slide. So this is something related to, summarizing what we have actually seen. A couple of these are the examples around what happens when we do with a stable pool strategy. Typically, the stable pool strategy takes around 6 to 8 weeks time to trigger that. And then if we would have gone in a conventional way, it would have taken around 20 to 24 weeks. Up to 16 weeks of time is saved towards starting the process development work. Then comes the early platform analytics. What it allows us is here in the upstream platform analytics is that we can do a risk assessment well on, and every stage has undergone a risk assessment so that we can take early decisions on that. So this helps us in actually augmenting any kind of difficulties that could potentially come with a specific molecule or specific biologics. Formulation, timing the formulation is very, very important because it's just inception and waiting. That would not solve the purpose of the encashing on the opportunity. So we have all of these platform analytics, platform processes, formulation. They have to be platform and they have to be timed very strategically. And as I said, any of these outsourced services, in this case, we have given an example of MCB and virus clearance study. How do we augment and move seamlessly from the development to the virus clearance services? How we augment so that we don't have to wait for getting the results. However, all the regulatory results would basically follow on as we actually, in parallel, as we move on into the project. So again, this is almost on the similar lines as I discussed over here. Some of the compared a little bit on the traditional model and the accelerated model. As we said that the stable pool typically begins, in case of a traditional model, between 20 to 24 weeks. This is what has been seen. We have seen we start the process development work in the 6 to 8 weeks when it is an accelerated model. Then, of course, the analytics, the CQAs, which we basically do a paperwork on the CQAs, run those CQAs in the accelerated model well ahead of time. Of course, in case of a traditional model, we wait for some outcomes that would basically define how good we are on the CQA, apart from the paper definitions that we have. Formulation, again, starting the formulation, we discussed it. And again, we about the MCB and the virus clearance, which are outsourced services. We said it, how do we de-risk it? So how do we manage the risk is the key messages here. When we do a parallel clone screening, platform methods, scale down de-risking, and regulatory comparability databases based upon the in-house informations that we have. So we then move the project very confidently ahead of time. And in case there are certain unexpected outcomes that we start seeing, we have time to take an action. So what we defined it as a fast-track therapy necessarily does not mean cutting corners, but it is an opportunity for various reasons, what FDA has mentioned. But how do we take this opportunity is by this augmented approach. Key takeaways, very important. There are something which is on the technical hands, and there are something which is logistical in nature. So strategic foundation for any of these fast track development, for fast track therapy, or even if I call it as a quicker development, the speed in the development are platform centric strategies. Every modules, every development work should have a reference point to start, which we call it as a platform, and that should be robust enough across various molecules. For this, of course, we talked about the antibody. Then we can make an acceleration based upon the capabilities that we have. Now, the thing is that, organization to organization capabilities varies, so it would not be that the same type of the strategy would work in other organization. It might need some customization. Remember one thing, that that's the reason we are saying it, that one size would not fit all type of thing. And the most important thing is that discussion within the teams. It is a collaborative ecosystem. We have a significant amount of discussions that happens on the paper, through the meetings within the team, and it's a huge amount of a cross-functional collaboration, not only the technical team alone, but the regulatory team with the quality operations. From the operation point of view, as we actually execute this project, there are proactive risk managements that needs to be taken care. Parallel pathing, of course, we discussed that. And based upon the data, we do a de-risking by comparing at each stage how the molecule is behaving, how the molecule is moving forward. So I think this is all I have from my side. I would be happy to take questions towards the later part of it, but I will hand over the baton to my colleague, Abhishek Bhatt, who will a little bit deep dive into the different top part of these fast track therapy as well as speed in execution. Thank you, everyone.
Yeah, so thank you so much and good evening to everyone. So I'll be diving into the section where we will be discussing about that what is the process of shifting to an integral model and how it works and how it helps. So first of all, I will be discussing about the overview. I will be discussing about the approach. I will be discussing about the strategies, success factors, and the key metrics that makes this integral model to achieve this kind of accelerated targets. So first of all, to discuss about the overview. So the very first aspect of this particular integrated model shifting is the fast track challenge. So as we all know, that the clinical timelines becomes drastically compressed as soon as the fast track designation happens, and the CMC becomes a rate limiting bottleneck. So these are the challenges we have to follow and overcome. Now, the next thing is that the traditional scale up and sequential development from R&D to pilot to the GMP also creates a lot of unacceptable delays, which require and this whole scheme requires a lot of risk-based decision making and constant streamlining. Now, the next aspect is the core paradigm shift, which is which that we have to adapt and implement, on the basis of the molecule robustness and the impurity profiles and all the aspects. That first thing is platform driven development, in which we can implement standardized 4 to 6 purification template depending upon the needs of the molecule and the impurity profile. Then next thing that can really help is the high throughput screening, which gives us an confidence to micro scale to screen out the micro scale resin, buffer mapping, and other aspects, so that we can screen out the most favorable outcomes and the working resin. Next is process intensification. Continuous process monitoring and the intensification gives us a great lead in order to increase a good amount of throughput and protein recovery throughout the process up to the drug substance level. And single-use technologies creates is a big game changer, because it gives us it enables us to avoid a lot of cleaning validation that creates a lot of intermediate and in-process delays. Now, the third thing is the integrated execution, what it means. So the parallel tech transfer, which is very important in case of a CDMO, and especially if the receiving and sender sides are different or are not in the same vicinity. So the CDMO presence should be from the very day from the very day one of the process development, and even from the cell line development stage as well, which is a great thing about Aragen, that we have both the things in the same umbrella. Now, the next thing is rolling BLA reviews. The incremental Module 3 submission to FDA plays a great role in case of a fast track submission that enables us to address the queries proactively. Now, the stage gate validation, which is scale down models with a qualified and established scale down model, we can predict a lot of process trends, and we can also predict a lot of projections and the profiles of the process up to the highest GMP scale, in which is 2 KL in our case. And last but not the least, raw material and consumable streamlining. In terms of logistics or qualification or even the dual sourcing, all these aspects are very important if not taken care in the on priority, which can create a huge bottleneck during as soon as we will reach to the GMP stage. So considering all this overview about the things, I would like to go for the approaches that we can follow to adapt and implement an integrated model. So the first thing, as we have briefly discussed, is the standardized platform templates. How it helps? We can utilize the proven platforms and modalities like protein A affinity and the viral inactivation steps, and other primary and secondary polishing steps like anion exchange, cation exchange, and the other filtration and processing templates, which are proven with a good amount of productivity. Then the next thing that can help us a lot is to minimize the customization. So if our molecule robustness allows and our impurity profiles are well into the controllable limits, then we can avoid any kind of any introduction of complex modalities, which can create a much more delay in development from early to the late stage phases. Now, the next thing that is very beneficial is to go for the high throughput screening, which gives us a very quick results in terms of screening things which is working for the molecule process development and which ones are not. So these kind of very quick resolutions gives us a chance to select the best ones in a very least amount of time, and create filling the gap between the timelines that comes between the early stage and the late stage of the. Now, the core principles that are very important to follow these approaches and to implement these approaches, I would like to discuss about those. And the first one is the well-defined step sequence. So this particular core principle, if we are following it, it can enable us to provide and to implement the already established platforms for multiple map candidates. Yes, it also, again, I would like to mention that it also depends on the robustness of the molecule. But if we are able to establish a single platform, though we at least we have something to start with for other map candidates also. Then the next thing is, which is very crucial and the backbone of any purification process, is the standardized buffer matrices. It restricts to choices to pre-qualified system, and it also helps us to maintain the some standard buffer backgrounds like citrate, phosphate, acetate, and trace, so that we can try a combination of these backgrounds and achieve our favorable process outcomes and everything. And not just that, not just apart from the process benefits, it also gives us an advantage to have advanced, it reduce the raw material qualification and assay development in the later stage. Now, the next thing is about the pre-pack columns. Up to a certain stage of development, if we go with the pre-pack columns, it also reduces our time, resource, and other things for the packing activities and validation activities of the column, and also gives a very representable process which can be scaled up further. And the very important thing is the fixed platform metrics. We have to be very clear about the in-process attributes as well as the final attributes, which should be within the regulatory acceptable ranges, and then we can have a confidence over, and we can have a decision tree over the things, over the processes. Now, after implementation, and if and the implementation and success of the approach, then we have to go for the different technology transfer strategy. And here I have compared both the ways to tech transfer, which is like traditional as well as the fast track. And here I have compared on the basis of execution model, facility fit, comparability, and validation. So if I talk about the execution model, in a traditional manner where it is sequential, the handoff from R&D to pilot scale to GMP, so it can create a lot of delays. And I'm not saying it's not a efficient method, but considering a accelerated target, it can create some delays which cannot be handled quickly and cannot be resolved. But if we go to the integrated fast track approach and strategy, so parallelly joint teams can work in a collaborative manner, and the CDMO involvement in case us, if it is from the very day one of the process, from the cell line development till the process development to the scale up, then it creates a very collaborative environment, and we can sort out the things, resolve the things in a very quick manner. Now, the facility fit, which in case of a traditional method where the facilities sender and receiver unit are very apart from each other and uh, don't have that connectivity, so the retrofit creates a lot of delays and kind of gaps. So whereas if we go to the fast track integrated strategy, we'll clearly see that the if we can understand the process from the day one, and can resolve the things in advance in our facilities and in the receiving unit, so that so it will benefit a lot, and our facility will be enabled in advance prior to the GMP batch. Then comparability, which is one of the most critical aspect. Then extensive side by side and analytical testing can create can cause a lot of delays, and some kind of gaps can be there. But when it is a risk based and kind of streamlined platform approach for through attributable methods and platform assays, then it can also helps us to get ready for in prior to any GMP batch. Then validation is also one of the thing, and which traditionally we can follow through the three sequential full scale commercial validation batches. But in a fully integrated fast track approach, we can use stage gate PPQ approach, where the qualified and evaluated scale down model can be very beneficial for that. So the key impact of all these integrated tech transfer, which we should understand, is that if we embed these kind of strategies for an accelerated target, then it creates a lot of it fills a lot of gaps on priority, and there will be no issues like retrofit of the facility or any kind of troubleshooting, and we can pull down the duration from 18 to 24 months from lab to GMP. Down We can pull it down to the 9 to 12 months span, which we have achieved in our case. So now, after understanding the overview of all the challenges and then approaches and strategies for development and tech transfer, it is very important to understand that what are the success factors of a tech transfer. So first thing is the scale down models. So the bench scale down models, which works in between the 1 to 5 ml, and sometimes it's 20 ml bench scale, it gives us a very closely mimicking process which mimics the commercial fluid dynamics and profiles, and even the quality attributes as well. We can have a predictive performance through these scaled down models, we can predict, we can kind of project, some projections can be made and forecasts can be made that what can happen at the highest GMP scale, like 2 KL or so. And this gives us a very close idea so that we can be ready and we can troubleshoot. And when we say troubleshoot, these scaled down models also help in root cause analysis. If there is something in the intermediate scale of where we are at 50 liter or 200 liter, and we face any kind of trouble in terms of some process parameter or some process, so we can troubleshoot these things down by using a SDM or a scale down model without investing a lot of resources and investing a lot of resources and time. So this plays a very crucial role for a success factor of tech transfer. Now, the next thing, very important thing and very collaborative thing is process description and tech transfer docs. These documents create a streamline between the multiple CFTs and create a proper communication among them. So how it helps, it also give a clear operational range and distinguishes between the normal operating range and the acceptable range, which is very crucial when you are operating and you have to focus on on some parameters that comes under this category. Then, as I already said, it creates a kind of platform, a bridge of a seamless communication, so that every single information is shared between among all the R&D stakeholders to the pilot scale stakeholders as well as the GMP stakeholders. Now, last but not the least, supply chain alignment, as I briefly discussed about it earlier, that raw material and the consumables, their logistics, qualification, and their procurement also is a very big bottleneck and can cause a lot of uh, can create a lot of hurdles in this entire accelerated target achievement. So the strategy that we can use can be dual sourcing strategy, where we can qualify multiple vendors for the same grade material, where we can raw material qualification, we can do the qualification of raw materials, especially the critical ones that will be in contact with the formulated drug substance. Then single use standard, where we can standardize the make MOC of all the tubings, bags, containers, and connectors across the sites, whether it is sender side or receiver side or anything. So these success factors, by applying and getting favorable outcomes out of all these steps, these are the success factors that proves that the tech transfer was smooth, and now we are ready, or we have already completed a successful batch. Now, after implementation and adapting all these particular strategies, now should be some key data metrics that are required for fast track CMC submissions. And these are the indicative of a successful submission as well. Like when we already have a clearance study of impurity and viral both in picture, where for the phase 1, we will do for the 2 panel virus, which will be retrovirus and parvovirus. Like we have whole time and stability session validation we have for not just for the end product, but also for the intermediate pools and tested pools also, where we have leachable and extractable studies also, where we can do a risk assessment for single use bags and all other consumables which are critical in nature, and leach protein A, which is a very critical process related impurity from our very first affinity step. Finally, it is product comparability profile. So pre versus post transfer equivalency across the scales from the R&D scale to pilot to GMP. The comparability gives us a actual picture that how successful our transfer is, and by applying the integrated approach, the risk-based approach, we can do it and in a very minimalistic timeline to achieve that accelerated target and to achieve this fast track monoclonal antibody submission. So with this, I thank all of you. And this is all I have to share with you about my understanding and experiences about our current achievement. And I would like to request Mr. Prithvish to take over and take us through the next step of our presentation. Thank you so much.
Yeah, thank you, Abhishek. And so today I'm going to talk about the FDA Fast-Track MAP program through the integrated approach, combining the technology transfer, process intensification, and cost optimization while maintaining the product quality. Here, the speed is not the objective. Speed with quality, scalability, and the COGS improvement is the objective. There is a saying in the biopharma manufacturing, the first gram proves the science, but the next thousand of grams prove the process. So with that, moving to the next slide. Let's start with the technology transfer because most manufacturing success or failure starts here. So this slide shows the confidence assessment framework. We do not treat every technology transfer the same. We classify the project as low, medium, or high confidence based upon the process understanding, platform fit, and the development history. For the high confidence project, we can accelerate aggressively. For the low confidence project, we perform a risk assessment and targeted development before the scale up. The risk-based approach compresses timeline without increasing the risk. We understand not every project needs more development, but every project needs the right development. So with that, moving to the next slide on the evaluation workflow. Our workflow is simple. Review the transfer package, identify gaps, assess the risk, and decide whether additional work is really necessary. The fast track development does not mean skipping steps, it means eliminating unnecessary steps. Once a technology transfer package arrives from a client, our first objective is to understand whether we have sufficient knowledge to move directly into the manufacturing readiness or whether we need to additional development work. We start by reviewing the technology transfer dossier and extracting all critical operating parameters across upstream, downstream, and analytical activities. The next step is identifying the information gap. In many cases, missing information is not necessarily a problem, but identifying the missing information can become a major risk during the scale-up. We then perform a structured risk assessment and ask some key questions. Is the process scalable? Is the characterization sufficient? Are the analytical methods fit for the purpose? Is the process compatible with the manufacturing platform? Are there any risk to the product quality, yield, or the timeline? If the available information is adequate, we move directly into the technology transfer planning, bomb preparation, batch record preparation, and manufacturing readiness activities. However, if critical information is missing, we perform a focused experimental work. This may include method qualification, small-scale confirmation studies, process verification experiment, or targeted process optimization. Here, the objective is not to do more experiment. The objective is do only the experiment that reduce the manufacturing risks. For example, if a client provides the robust scale-down model data, process characterization data, and previous manufacturing history, we may confidently move directly towards the transfer execution. On the other hand, if we receive excellent laboratory data but the limited information about scale-up performance, we may conduct targeted verification studies before committing to a GMP manufacturing. So with that, I wanted to showcase the case study, what we have performed here at Aragen and deliver a FDA fast-track project within 9 months. This slide shows up to this point, I have discussed our technology transfer philosophy and decision-making framework. Let me now show how this translated into a real-world monoclonal antibody program. This program was operating under an accelerated timeline, which means that we had to balance speed, technical rigor, and the manufacturing readiness simultaneously. As shown here, the journey started with the cell bank receipt and the MC preparation. Rather than waiting for every activity to be completed sequentially, multiple activities were executed in parallel wherever the scientifically justified. We initiate the technology transfer activities in AMBER 250 and 10-liter bioreactors, rapidly generated the process understanding, and used those learning to log the process before scale-up. The process then progressed through the 50-liter, 200-liter, and ultimately 2 kL GMP manufacturing scale. This development pathway from the cell bank received to the 2 kL manufacturing was completed within a highly compressed timeline. The reason this was possible is because We used a risk-based approach rather than a sequential approach. So the achievement I am most proud of is not the 2 kL batch itself, which we delivered, but we have done a consistent delivery across the scale from the amber to the 2 kL throughout amber, 10 liter, 50 liter, 200 liter. We have seen the similar performance across the scale. So with this, I'll just go to little off tracking, because with the FDA fast tracking, it does not mean that we just deliver the project within the time. It is also mean that that how cost effective we are, and how we can bring more productivity into our system. With that, I'll focus more on the process intensification from my next slide. Yeah. So up to this point, we have discussed how to transfer a process successfully and rapidly. The next challenge is equally important. Can we significantly improve the productivity while maintaining the manufacturability? In today's biologics landscape, tighter alone is not enough. Companies are looking for shorter timeline, lower manufacturing cost, and a higher facility utilization. Process intensification is our approach to achieving these goals while maintaining the product quality. For the fast track program, it is also important that we set a tighter expectation and a COGS expectation clear from the beginning. Fast tracking does not mean that we compromise productivity and COGS. In my this slide and upcoming slide, I'll take you through some of the case studies what we have done at Aragen end, not necessarily for the new modalities, but also with the biosimilars, that how we have able to improve or apply this process intensification strategy and move ahead with the better outcome on the product, on the titer and the COGS. So this slide shows about the metabolic insight-driven process development. Traditionally, media and feed optimization relied heavily on trial and error experiment. Multiple conditions would be tested, and improvement were often discovered experimentally. Today, we take a much more a data-driven approach by understanding cellular metabolism and the nutrient utilization pattern throughout the culture process. By monitoring which nutrients are consumed, depleted, or accumulated at different stages of the process, we can identify the metabolic bottlenecks much earlier. This helps us actually the design more targeted on the media feed strategies rather than relying on the extensive screening campaigns. For example, if a key nutrient becomes limiting during the production phase, we can address that productivity through the feed design rather than discovering the issue after multiple rounds of development. The benefit is not only the higher productivity, but also faster process development timelines, reduce experiment, and better process robustness. We are moving from the trial and error development to the knowledge-driven development, and this metabolic mix analysis are actually helps in this space. So in my next slide, I'll be showing that how we would be able to, means how we have been achieved at Aragen with a higher titer by keeping the media consumption and also the viable cell concentration lower, and also by improving the PCT. After understanding the metabolic requirement of the culture, we asked a practical question: can we improve a concentrated fed batch process using insight from the spent medium analysis? We performed spent medium analysis and identify the nutrient that are being consumed rapidly within 45 the process media using component which consume faster. The outcome was significant. We achieved approximately 17 gram per liter of the titer by day 10. We maintained a consistent QP of approximately 40 PCT, and importantly, the media utilization remained efficient. The goal here, I wanted to emphasize, the goal was not to maximum the titer. The goal was maximum the productivity with control media cost and operational flexibility. The next slide shows that how with this approach, we are able to take up a titer from the fed batch to the concentrated fed batch up to the 3x improvement, where we have achieved a titer of the 27 gram per liter. Also, the next case studies, moving to the next case studies, we have multiple case studies on that. Yeah, this next case study shows that how we have performed a same molecule in the different uh, uh, process, like the fed batch, high seed density batch, and the concentrated fed batch, where the fed batch yielding around 1x, high HSD yielding around 1.5x, and the concentrated fed batch yielding more than 3x of the titer. So with the enhance of the titer, the important message here is to reduce the COGS. The next slide shows the same, how we can, because it's the most important parameter of what we should achieve at the end. This is for ultimately the patient do not buy the titers. Companies do not sell the cell densities. Companies sell medicines with the profit. So when we analyze the typical 2-kil fed batch process, media and feed represent approximately 38% of the total manufacturing cost. So we immediately identifies that largest opportunity for the improvement. So if we run some kind of with this increasing titer, with the reducing media consumption and optimizing the filtration strategies and reducing the downstream disposable cost, we can reduce this COGS further. So in this slide, we have shown that how the titer can reduce the COGS from in every gram of the increasing of the titer can reduce the COGS significantly, and we would be able to, means at Aragen, we are able to achieve a COGS of below $70 per gram. So we are aiming for further lower with our optimized process, and also the understanding from the metabolomics and the spent medium analysis. Moving to the next slide is that it is not only the increase of the titer and the COGS reduction, we also equally focus on the product quality improvement. And this slide shows that how we have developed a model for the statistical model for the glycosylation improvement. This molecule shows where is that the yellow bar shows the innovator glycan pattern, and the brown shows the titer, means glycan pattern for our process. But with the intensification and with this statistical tool, we are even able to able to match the product quality, which shown to my next slide. So it is a transformational thing from the product quality being a sub-optimized from the innovator to a very optimized product quality using the statistical model for the process quality development. In my next slide also shows that with the intensification process, our how the attributes such as G0F, galactosylation, and the terminal glucosamine remain within the expected range. And not only with this bar diagram, we also have done the statistical analysis to show that how comparable the product quality, and also they are not statistically insignificant. So moving towards the next slide shows the statistical data done with the different product quality attributes, shows at a different scale with the different with the process intensification approach, we are able to able to maintain the product quality as per the innovator. So I'll not much take my time on this slide. So this slide shows the alpha-glycylation pattern. The next slide shows the galactosylation pattern. The next slide shows the sialation pattern, and the next slide shows the G0F pattern. So it is not only with this traditional approach, and but also with the statistical approach, we are able to prove that there is no significant difference between the intensified process and the normal process from the innovator lot. So with that, I wanted to close my presentation with three takeaways. First takeaway is that the technology transfer should be a risk-based. Not every program requires the same development strategy. Takeaway two is that the process intensification can deliver multi-fold productivity improvements. Takeaway three is that productivity improvements should translate into reduced cost per gram while maintaining the product quality. Historically, development teams believed that they had to choose between the speed, productivity, quality, and cost. Our experience suggests otherwise. With the right technology transfer framework, data-driven development strategy, and process intensification approach, all four objectives can be achieved simultaneously. So, with that, I'll end my presentation, and I'll thanks to all the audience for listening to me. And if there is any question, I'll take it up after closing the webinar. With that note, I'm now handing over to Gokul Ram to take us through the analytical method transfer and regulatory audit roadmap. Thank you. Over to you, Gokul.
Thank you, Prudvesh. Thank you for handing over to me. Hello, everyone. So I'll quickly walk us through the analytical tech transfers and the regulatory pathways that we have adopted for the recent FDA Fast-Track application that we have done, and we have successfully completed the IND submission. So here is the integrated timeline, what QC does, and how did the method transfer and the Fast-Track designation has happened to compress the CMC timeline. We have fully integrated into our ICH Q2R2 and Q14 aligned validation and Q5E anchored comparability for all the activities that we have performed, right from the process development to tech transfers to scale up to final finished product and the quality attributes, critical quality attributes. We have identified some of the risk categories and the potential impact it can pose, and we have mitigated those risks with a strategy that we have evolved in fast track designation. First and foremost is the parallel pathway of method transfers. If it happens a sequential way, it will lead to a fatal time delay. So what we have done is all the method developments, transfer protocols, all of them have been drafted in unanimous, parallel pathways for the PD and MSAT to work on the process, while the analytics have been working on the platforms that have been designed and validated and successfully transferred to our QC. While the tech transfer is happening to QC, what are the challenges that can happen at the site where the transfers are being done? What is the robustness of the method is very important. What are the risks involved in an unsuccessful transfer? And it can lead to some of the OS that are being contributed from the analytical methods rather than from the process. So to minimize that, as full-fledged validation protocol as per the ICH Q2R2 has been explicitly designed, and the variables that are related to stress studies and a thorough assessment of risk have been evaluated from the originating lab and have been successfully evaluated, and reproducibilities have been demonstrated at the receiving labs, that is at the QC labs. As our colleague mentioned earlier, some of the comparability runs have been developed for all the batches that have been anchored right from the 50 liter, the 200 liter, the 2KL. All have they been compared. As my colleague Prudvish was mentioning, we have done a full-fledged critical quality attribute panel, the charge variants, the glycan profiles, the potency of the all the batches that have been manufactured, and all the scales in the host cell proteins and the host cell DNA. All of them have been thoroughly evaluated as a comparability profile, and the critical quality attributes have been successfully compared for the IND submission, which is very, very important for an FDA auditor or an FDA evaluator. How did the scale up dynamics happen, and what is the final quality? Is it comparable to your scale down model? All of them have been thoroughly evaluated. So another risk that can potentially happen, as I discussed earlier, we can run into OS and OT if the right method transfer doesn't happen. So a scientific, thorough 6M and 5Y analysis to distinguish an assignable root cause, put in place the CAPAs that are required to improve the methods. All of them have been identified right from the day one, and have been designed well in the method development, method qualification, and validation program. Another important aspect that as we scale up and move into a GMP environment, bioburden, endotoxin, environmental control rigor is very, very important. Otherwise, it can potentially lead to batch contamination or a bioburden at a final stage, which is a fatal thing that can happen to a drug substance or a drug product or a high and elevated endotoxins that are potentially risk contaminants to the patient safety, right? So to minimize all these things, we have done a full-fledged, uh, contamination control strategy, and the methods have been redeveloped, and endotoxin have been thoroughly evaluated for all the drug substance and drug products that we have manufactured, which would help us in fast tracking these products. A thorough EU Annex 1 and FDA 1116 chapter risk assessment strategy has helped us identify the risks and mitigate all of them. With these designations, we have done a successful IND submission in less than 9 months, is what our track record has been, which has been a significant track record in the industry to file a FDA. Moving on quickly to the regulatory pathway, Aragen's capabilities on regulatory pathways have been phenomenal. We have had several FDA audits and EMA audits and PMDA audits successfully completed across the various units of our organization. And at biologics, we have completed successfully the IND submission, and FDA BLA applications are going to happen. And the quality system remains at a corporate level. Some of the digitalization platforms, the LMS, UMS, QMS, EDMS, and the LIMS, all of them are digital, and the global regulatory pathways have been thoroughly evaluated by FDA and EMA, and they have been evaluated and successfully we have completed the FDA inspections without any zero observations, and we have received the EIRs in all the units where we faced FDA. So this is our global regulatory footprint while we have gone on to the pathway for accelerating our submissions for FDA. So I'll take a pause here. I'll open up the questions. The forum is open for questions. This is the last slide, and I will leave it to the host to give us some questions. Thank you.
Thank you to all our speakers, Indranil, Prithvish, Gokul, and Abhishek, for sharing your expertise across the biologics development life cycle, from process and analytical development to technology transfer, and including manufacturing quality and regulatory readiness. Now we will move into our Q&A session, and I can see like a lot of questions are coming in. Given the time, we won't be able to take all of them, but Indranil sir, we have this first question. So uh, the audience wants to know that given the industry's long-standing focus on improving titer, should we still consider it as the most important metric? How you will take this?
So it varies depending upon what the strategy is around that. Typically, for early clinical phase, it may not meet, but for the late phase or existing asset, it do mean, of course, keeping the quality into consideration. So it is on a kind of basis of what's the kind of a requirement that is there. So I would say that it can be yes and no in this case.
Yeah, thank you, thank you. So moving to our next question, it says that I think like Prithvish, it is for you. So Ashiana is like she wants to know like if a client approaches Aragen with the process developed on a completely different platform, would you still recommend a fast track transfer approach?
This again depends upon that where the process have been or how the process have been developed and how fast the client wanted to go to the market. A fast tracking, yes, obviously help if there is a requirement. And whatever the platform it is, there the strategy of the risk assessment and the gap assessment at the initial phase takes the major role, where we can identify the gaps. If with the different platform, can you adapt to the Aragen platform, and what is the time or what is the level of experimentation you wanted to have? If the answer is yes, we can do with a minimal, minimal experimentation. Why not? Why can't we do a fast tracking thing? We can always move if we can deliver consistently with the higher productivity, quality, and in time. That is the focus at Aragen from our side.
So there is one more question we have, Prithvish. You have been telling us that beside, like, it's not only about the timeline, quality and COGS also is something which we need to consider. So, given that fact, like, where do you see the biggest opportunity for future COGS reduction? Because you highlighted that media and feed are the largest contributor to manufacturing cost. So, what is your viewpoint in that?
Right. So, media and feed is another line of approach. The major contributor for the COGS reduction, what I wanted to mention is that definitely the increasing the productivity, decreasing the cycle time, and decreasing the facility utilization. So the world is moving towards, that majorly the CDMO players are playing a major role for developing the program for different client. So the speed here is the essence, and with the process intensification approach, it is important that how you can reduce the turnaround time. The success lies there because most of the COGS are involved with your facility utilization. So that something should be bring down significantly. Along with that, the scientific approach on the process intensification by reducing the media and feed, and also the single use items, which are the main contributor to the COGS.
Great, great. So next question is, so Abhishek, you mentioned that one of the critical factor which we need to consider is standardized platform. So does standardized platform approach can be followed for every molecule?
Yeah, so what I briefly also discussed during the presentation as well. No, it's not necessary that the platform will be successful for every map candidate or every protein candidate. But yes, having a platform in place can give us something to start with. And it majorly depends on the molecule robustness and the impurity profile. So if my molecule is robust and I am able to control all the impurities through a platform process itself, so it will give me a very expedited process. But if my molecule itself is a little complex one with different product related impurities, then it takes a little different. We have to go a little out of the box. We have to go for the multimodal modalities, like those things. So in that case, yes, it will give us a kickstart, but it will not give us a complete success. So yes, platform process is a very good tool, but it's not necessary it works every time.
Thank you, Abhishek. So, next question is, Gokul, it's for you. How do you handle OS and OOT investigations without derailing an accelerated stability program?
Thanks. That's a wonderful question. So typically we see OS and OT at some of the accelerated conditions because biologics are not like small molecules. The Arrhenius equations for extrapolation of biomolecules doesn't work the same way as the smaller molecules. So it becomes very tough to predict the modeling for next time point, what would be the kinetics of some of the critical quality attributes that we discussed, like charge variants or probably the potency of the drug, or some of the critical quality attributes that we have identified during our scale down model or early regulatory pathways. So it becomes very challenging when you have only one or two batches to determine the shelf life at this stage of the stability studies. So it needs a thorough risk assessment, an OS and OOT investigation right from the phase one as per the MHRA guidelines. The phase one should lead us that there are no assignable causes from the laboratories. There are no contributing factors from any of the instruments or reproducibility of the methods or analyst. So you should rule out all these errors that can potentially happen as an OSOT investigation in the lab, and it should actually lead you to a product profile or a typical product being failed at a later point of time, as per the you know trends that are sequentially or evaluated to a gradually increase in the trend, which should lead you to an OS and OOT. A sudden spike should not happen in any of the stability studies. It's not a healthy pathway to evaluate an OS and OOT.
Yes, definitely, Gokul. I agree. So, analytical development is something which is very important, as Indranil sir also showed us in his timeline that how we started early. But Indranil sir, I want to we have been asked by the audience. Samuel wants to know how early should analytical development begin in an accelerated program? Maybe Gokul, you can also add to what Sir has to say.
So there are two aspects into it. There is a aspect. What we typically do in a program is that the early stable pool generation materials actually gets into the process development. As a result, it generates material good enough fit to start on the development work. We segregate between process related impurities development and product related impurities. We start with the process related impurities well ahead of time because we do a platform process assessment how the clearance of the process related impurities are happening. Product related impurities are mainly related to the product stuff. It happens at a slightly later. Having said that, those product related impurities typically are taken when the downstream process is slightly more matured, say around 6 to 8 weeks of time. That's where we basically start working on the product related impurities. So the qualification in the meantime starts for the process related impurities, follow it up with the product related impurities, because by that time there will be some representative samples wherein it needs qualification, needs some representative samples so that they fix up dilutions, fix up the various parameters associated with the qualification. So in short, these are the typical timelines that would require both for development and qualification.
So
Gokul, you
Gokul,
want to add on anything? Yeah.
I know, I think, Indranil, you covered everything. I just want to add one point. It's very important for method development to happen in a very robust manner, because we cannot go back in time and redevelop the method. That's a risk to the entire program and the process. So a method robustness should be thoroughly evaluated while we are developing the method. And it's very, very important to have a platform method. That's why Aragen's platform methods work wonderfully for most of the molecules that we have evaluated. And this is where we are very strong at an analytical background, and we have developed and established and thoroughly evaluated most of the molecules available in the market. And we have done it in very minimal time to qualify and validate all of our platform methods.
that's great. So that is all the time we have for questions today. Thank you to all the speaker and our audience for today's session. If your question wasn't answered or if you have additional queries later on, please feel free to reach out us to us at info@aragen.com. Thank you all for your active participation and for joining us today. Have a wonderful time ahead, and we look forward to connecting with you again soon for our next webinar on another engaging topic. So stay tuned. Thank you, everyone.